going to dust off my undergraduate lecture from when I was a professor, adjunct professor at Ohio State, and taught an introduction to virology a long time ago. So I'll walk you through a little bit what viruses are, what happens when viruses infect people, and it'll set the stage for discussions about how we're thinking about dealing with the COVID-19 crisis, both with vaccines and therapeutics. By now, all of you probably have seen and know what a coronavirus looks like schematically. So, and the key thing here is to give you a little bit of a picture about what viruses are. So they are sort of in between living and inanimate. In their native state, when they're not in a host causing disease or growing or replicating, they're simple particles of protein and nucleic acid and lipid. And basically what they are is, if you look at this diagram here, they have a core of a nucleic acid, which we know of as DNA and RNA. In this case, the core is RNA, which is the blueprint for manufacturing new virus particles. And basically the rest of the particle's purpose is to deliver that piece of nucleic acid to a cell where it can be used to replicate and make new copies of the virus. So that nucleic acid is encased in a lipid or fat envelope, which is shown there in red, and it has embedded in that envelope these spike proteins, which look like inverted coronavirus bottles in this case, and other proteins. And so the spike protein that you see there in yellow is what allows a virus to attach to a cell and be taken up by a cell and to begin the process of replicating and actually, turning on that blueprint, that algorithm that's encoded in the RNA there. So if we go to the next slide, I'll give you a schematic overview of the life cycle of virus replication. So the virus attaches to receptors on the surface of cells, in this case, angiotensin converting enzyme receptors. Not important that it's a protein found on the membranes in the lining of your throat and nose. So it's basically pulled into the cell in a membrane, and the contents of the virus are released. So the RNA is released, and that RNA instructs the cell so that the virus comes in and takes over the replication and protein manufacturing machinery of the cell and turns the cell into a virus-making factory. And so that blueprint's released, and it is translated into protein pieces that both are used to assemble new virus particles and to replicate or make copies of the RNA to be incorporated into those particles and then released again from the cell, either by budding out of the cell or by the cell literally exploding and releasing those particles to spread and affect surrounding cells. So that's the life cycle. And I think the point here is you see there's a number of steps that involve release and protein translation and replication of RNA and the actual attachment process. All of those are targets for therapeutic intervention, either by vaccines or new drugs. And Tim will probably get into that a little bit more, and we can talk about that in more in detail depending on the questions we get. So if we go one more slide to sort of complete the process, this is what a virus is doing, and this is what you are doing when you're infected with a virus. The virus begins replication, and immediately the host begins to respond with an immune response. Now that takes a bit of time to build up. So what happens is cells in your blood called B cells recognize the virus particles as foreign and begin to make antibodies. And they make two classes of antibodies, IgM and IgG. IgM comes up in the first 7 days and begins to decline. IgG comes up after that and is in much larger amounts. And the antibodies are the primary mechanism by which the body clears you of the virus. And so over the course of about 3 or 4 weeks, the antibody response comes up, clears the virus, it begins to decline. And at the end of that, the virus is gone, and you're sitting there with these B cells that are capable of responding to that virus if they ever see it again. So these are called memory B cells. And so if you have a second encounter with a coronavirus or any other virus, the body responds much more rapidly and produces antibodies at a much higher rate and rapidly clears that infection. So you are immune after that first viral infection. The immunity is mediated by those B cells, those memory B cells that make the new antibody. So the same process occurs. It will clear the virus quickly, and those memory cells can be maintained over life. In some cases, like you have with certain viral infections, and others, it doesn't last as long. But in most cases, it has a duration of several years. So that's the process of infection and our response to infection. And what we're trying to do with regard to vaccines is mimic this immune response. So what we're trying to do is administer to individuals either attenuated viruses that will infect and raise a response that will act against the virulent form of the virus, or administer pieces of that, or administer the instructions to your cells to make pieces of the virus, which you will raise an immune response to. So those are a quick overview of viruses, how they replicate, and how we respond to them.
John, thank you so much. And I bet not only myself, but I bet many of our listeners have kiddos who are at home, and we're all wishing that they were just listening to you, because that could have been a great education moment for all of us. I will remind people that this is recorded, so do feel free to share this with your kids and with your family. That was very helpful. John, if you don't mind diving in a bit, I know a lot of people are saying, well, how is this spread, and are starting to have a better understanding around how it's spread from person to person. But there was also the question around animals. Do you mind answering that a little bit about how viruses are spread with animals?
So it's not common, but it's how a lot of novel viruses are introduced to the human population. So there are coronaviruses that circulate in animal populations, and when we come in close contact with them, every once in a while, one has mutated away from its ability to grow only in the animal host and jump to humans. That's actually how influenza changes. It jumps from birds to humans, or birds to pigs to humans. So that's the viruses are circulating in those populations. Once in a while, they jump to ours, and depending on how we respond to them, they continue to circulate, or they continue to change to enable themselves to circulate. So flu mutates, changes year to year, that allows it to continue to circulate in our population. We don't know if that'll be the case with this coronavirus. Other coronaviruses are around and continue to circulate in our population as well.